started here to keep us on time. Today I'm going to be talking about the role of environmental heterogeneity in maintaining variation. And as evolutionary biologists, we think a lot about the maintenance of variation within populations. As we all know, this is a key component of the modern synthesis. It has a long history of both theoretical and empirical treatment, thinking about how, in particular, genetic variation is maintained. Surprisingly, there's actually been quite a lot less research done on the maintenance of phenotypic variation within populations. Sometimes the genetic variation leads to phenotypic variation, but not always. But the maintenance of phenotypic variation really poses a conundrum because selection should erode variation in fitness-related traits. So this is sort of the question here. There are a number of mechanisms that have been proposed that could be involved in maintaining variation within populations. So these include things like spatial and temporal fluctuating selection. So the idea that the optimal target is moving around in space or time, and so you never converge on a single optimum. Balancing selection, frequency-dependent selection, diversifying selection, or many people know this as bet hedging, and then non-selective processes like gene flow and genetic drift. And in particular, two of these processes, so spatial and temporal selection and diversifying selection, or bet hedging, explicitly involve the component that the environment is heterogeneous and unpredictable. So that's sort of specific into both of those processes. And so I just want to think for a minute about this role of unpredictable environments and uncertainty. And I think as evolutionary biologists, we don't think a lot about uncertainty. We tend to think of sort of deterministic processes. But we all know that both biotic and abiotic environments are not predictable, and there is a lot of uncertainty. And this is particularly true for plants that are sessile and are stuck in the environment in which they happen to germinate and have to deal with all of that uncertainty around them. And so the question for an organism really becomes, can the effects of uncertainty be mitigated? And really, fitness depends on the ability of an organism to deal with this. So I'm going to focus this talk on variation in an annual plant, so with a very constrained life cycle, where we can kind of get a handle on where the key times of the life cycle that this sort of uncertainty might be most important. And this could involve the timing of germination, which is often thought of as the most risky transition in a life cycle. Germinating in the fall or the spring, and this would really depend on the risk of overwintering successfully. And then flowering, and in this particular plant, I show Mimulus guttatus, an annual population of it here. There's strong end of life cycle due to the onset of drought. And the timing of drought, obviously, is not fully predictable. So there's uncertainty at the timing of germination and uncertainty at the timing of flowering. Okay, so that's really what I'm going to focus on here, is how those two aspects of uncertainty and how plants can deal with that and potentially mitigate those unpredictable environmental events. So we were out in the field in a population on Vancouver Island, British Columbia, and noticed that there were plants that germinated in both the spring and the fall. And this photograph on the left here are from germinants on April 4th in a single population. And we have this plant on the left that germinated in the fall, overwintered as a rosette, and has clearly grown and established. And then this tiny little cotyledon plant here on the right. And these were represented in good abundance of both types. Now, as I said, this is an annual plant with a strict end of growing season by the onset of drought. And so the question really is how these two plants kind of compete through the life cycle, what the fitness effects of the two are, and how they're managing to coexist in this single annual population. So more specifically, we wanted to know whether there was a genetic basis to the seasonal variation of germination timing. And then either way, whether it was genetic or not, what are the fitness consequences for this? How do these two plants of really different sizes compete? They've got about an 8-week max window from this time to complete their life cycle. And then do we see any differences in the offspring of these fall and spring plants? Any evidence for transgenerational plasticity or differences in fitness in the offspring? So this is the location of the population there. We would have loved to do this in the field. Really challenging to be out there at both fall and spring for germination. And so what we did was what we thought was the next best thing is we downloaded meteorological data from the site. We set up a growth chamber to monitor, to change in real time temperature, day length, and humidity to track this. And then we did the experiment in real time in simulated conditions in a growth chamber. We did this in a quantitative genetic framework, so we had 20 maternal families that then germinated in either fall or spring, and we could look at genetic variation and heritability for these traits. Okay, so what did we find? So here I show you the day of the experiment. So again, this is sort of tracking real time here. We accelerated winter a little bit, but we did have a long break where the plants were cold and dormant there. And I just show you the density of seeds that germinated. So you can see that germination was about equal in both seasons, and the pattern of germination was roughly similar. So there's this little extra blip here, but overall those distributions are not different. What I show you on the right are the family means. And you can see that overall there's really not a strong effect of season. So we do have this one family that seemed to show cold induced dormancy, so it had much lower germination in the spring. But when we remove that family, there's really no significant season effect or G by E effect here. So this is not a genetic trait. So individual families are equally likely to germinate in both the fall and the spring. Okay, so that was that first question. So now kind of moving on to some of the consequences for this in terms of how these plants compete. So we did find that fall plants begin flowering earlier in the orange here. So the onset of flowering is earlier for them. The spring plants have a much narrower window of the onset of flowering. It's a little bit delayed. So flowering time is affected by this. We find that the onset of death, as predicted, was highly synchronous for the two cohorts. So this is driven by the availability of water. It runs out, plants die. And what this means is that the flowering duration is really different. So this is, remember, just the onset of flowering, but these plants keep flowering throughout that time until senescence. So there's not really much potential here for assortative mating, if you're thinking of that, or other sort of cohort by cohort interactions. There really are very likely just mating freely amongst these two cohorts during this window right here. But certainly the fall plants flower for longer. Okay, so those are sort of phenotypic distributions. What about some of the genetic heritability components of this? So here again, I show you family means. You can see again, those fall plants flower earlier. So that's flowering time here in the fall and the spring. But what was really interesting was a complete absence of genetic variation in the spring, largely driven by the absence of phenotypic variation. So if you recall that previous figure of the distributions, really narrow onset of flowering time. So they have this really constrained growing season where these spring plants just rush to flower as quickly as they can. No phenotypic variation, no genetic variation. So a really strong G by E here, almost entirely driven in unequal variances between the treatments, rather than classic crossing of reaction norms. Okay, so what effect does this have on fitness? So flowering time is one component of fitness. We measured fecundity, or flower number. So remember, these plants, at least at germination, are really different sizes. Sorry, at the start of the growing season. So when we look at flower number, there's actually very little effect on fecundity. So incredible variation among families in terms of how many flowers they make. So a lot of genetic variation for flower number, but no real difference between the seasons. On average, in fact, the fall and the spring make almost equivalent. Spring's a little bit lower, but not significant. So there really isn't a strong effect of germination season. Those spring plants are flowering quickly, but they're catching up in terms of flower number. So how do they do this? Well, we think one of the mechanisms is through alternative growth strategies. The fall plants have many more leaves than the spring plants, so they're sort of larger plants with a lot more leaves. But really surprisingly, we found that the spring plants actually have much larger leaves. And so what we think is happening here is that the spring plants are foregoing the construction costs of new leaves and instead are rapidly expanding the leaves they have, which is giving them enough photosynthetic energy to make the same number of flowers. Now, I think this is where that onset of drought might be really important. So in seasons where drought comes really late and you've got a longer growing season, it's possible that's when we would see the spring plants really benefiting of having more leaves, more time to expand them. But in a short growing season, that's not going to happen. Okay, so then we wanted to go on and look at the next generation. And so we did all round robin crosses here. So again, remember that they're flowering at the same time. So we crossed within and between all cohorts in all directions. So we had 4 different sets here, 40 crosses from each. We had seed with and without cold to kind of look if there was any transgenerational plasticity in whether they needed cold or difference. We also wanted to look at whether there were fitness differences just in terms of germination. So we didn't go on. We screened these all. There were 8,000 plants in a common environment, so this was a massive crossing experiment and then a massive amount of work looking at this offspring generation. And we didn't find anything. So this might look like really interesting crossing reaction norms here. Look at this axis. It's from 0.97 to 1. So everything germinated really quickly. So there's definitely no transgenerational plasticity in terms of what your parents experienced, what the seed experienced for cold. But there's also really no fitness consequences, at least in seed germination. So even those plants that flowered really quickly and didn't have a lot of resources, the seed are all germinating just fine. Of course, in the field, there's going to be other things in terms of pollinator visitation. We did find a strong treatment effect of germination time. That's been demonstrated before. Okay, so to wrap up here, it really looks like the variable life cycles is maintained by this germination timing, which we think is entirely stochastic and heterogeneous. So there isn't strong heritability for this trait. We think if a seed lands in a wet environment, it germinates in the fall. If it lands in if it doesn't, it waits until spring rains, germinates in the spring, and then has this plastic growth strategy to overcome that disadvantage to still make a fair number of flowers. And then so these variable environments is really maintaining this variation within the population. So it's a stochastic process. And then regardless of environment, total fitness is similar. But selection may not be able to act on those spring germinating plants in terms of flowering time because there was no genetic variation. So there might be really important consequences for how selection acts on this population given the different germination characteristics. Thank you. Yes, Linda.
I suspect there are other leaf differences as well, like leaf thickness or something about physiology. Do you think that might be?
Certainly, yes. So those larger, we did not measure it.
Yeah.
We did not measure other leaf traits. It's certainly possible that those larger leaves are thinner. Yeah. And I can talk to you that we think part of why the fall plants why they might be different is overwintering they might be investing in cold tolerance and possibly defense as well, that the spring plants are avoiding.